Good afternoon. I'm Dr. Kara Wilder, a technical writer at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series entitled The Biology of Anaerobic Bacteria and Predominant Propagation Practices, presented by Ms. Allison Faust and Ms. Nancy Kruger. Ms. Faust and Ms. Kruger are senior biologists at ATCC. They have extensive experience in the propagation and quality control testing of a vast array of microbial species and have participated in anaerobic workshops and internal anaerobic studies to determine the ideal propagation and test methods for anaerobes. In today's webinar, they will discuss the various methods used to achieve successful growth conditions for a wide variety of anaerobes. Here, they will consider topics such as common anaerobic gas mixtures, media selection, and obtaining anaerobic conditions in the lab. Taking into account individual laboratory constraints, they will also discuss several propagation methods available today and will use specific examples from the ATCC collection to demonstrate these techniques. If you have any questions for Ms. Faust or Ms. Kruger, please use the chat function available through the webinar program and direct your question to the speaker you wish to answer. Questions will be answered as time allows at the end of the presentation, and any remaining questions, as well as the recorded webinar presentation, will be archived on the ATCC website at www.atcc.org.
Thank you, Dr. Wilder. First, I would like to provide a bit of background on ATCC. Founded in 1925, ATCC is a private, independent, nonprofit organization with its headquarters in Manassas, Virginia, USA. ATCC is the world's premier biological materials resource and standards development organization that collaborates with and supports the scientific community with industry standard products and innovative solutions. Our broad range of products includes cell lines, microorganisms, genomic and synthetic nucleic acids, and reagents, with each of our biomaterials expertly authenticated and characterized through genotypic, phenotypic, and functional analysis. Our outline for today's presentation will involve an overview of ATCC's anaerobe collection, classification of anaerobic bacteria, specific examples of each class of anaerobes, nutritional and atmospheric requirements, and historic and current propagation methods of anaerobic bacteria. Currently, ATCC has 942 active anaerobe holdings, with 465 of these being type strains. This number does not include anaerobic extremophiles, which ATCC classifies as organisms that do not follow the established biochemical testing scheme for anaerobic bacteria. This group of anaerobic extremophiles includes archaea such as Methanogens, sulfate reducers such as disulfovibrio, and thermophiles like Pyrococcus. In the 1960s and 70s, ATCC's collection was fueled by the Anaerobe Laboratory at Virginia Polytechnic Institute, now Virginia Tech. Pioneers in anaerobic bacteriology such as W.E.C. Moore ran the labs at VPI. He perfected a way to routinely culture anaerobes using inert gases and helped develop a way to classify and identify strains and substrains of anaerobic bacteria using fermentation product patterns, enzymatic tests, and growth requirements. This eventually became the Anaerobe Laboratory Manual. These methods are the basis of modern biochemical assays.
The top 5 selling anaerobes at ATCC are Clostridium sporogenes, ATCC 11437, Clostridium sporogenes, ATCC 19404, Clostridium perfringens, ATCC 13124, Bacteroides fragilis, ATCC 25285, and Porphyromonas gingivalis, ATCC 33277. All of these strains are used in numerous testing assays for media, water, sterility, quality control, and validation of rapid microbial methods, or PCR-based methods, and antimicrobial susceptibility testing. Some of these items, such as 11437 and 19404, are available in multiple vial formats, such as easy-to-open serum vials and ready-to-use mini vials, to provide the customer the best platform for their needs. There are 3 different classes of anaerobes that are determined based on how the organism grows in the presence of oxygen. There are facultative, aerotolerant, and strict or obligate anaerobes. Please note that gram-negative facultative organisms such as Escherichia are not being considered in the facultative group for this presentation as their primary growth environment is aerobic. We will also discuss anaerobes found in the environment and extremophiles such as Methanogens. The growth and nutritional requirements of specific examples of these classes of anaerobic bacteria will be discussed in detail in the next slides. Facultative anaerobes can grow with or without the presence of oxygen. They can metabolize energy aerobically through respiration or anaerobically through fermentation. One example of this is Propionibacterium acnes ATCC 6919. Propionibacterium are non-motile gram-positive pleomorphic rods. This particular strain was isolated from facial acne and has variable aerotolerance. It grows somewhat in ambient air, but better anaerobically. When selecting a medium and atmosphere for an organism, specific nutritional requirements should be taken into account. Propionibacterium requires vitamin B5. This nutrient can be found in abundance in meat. Therefore, both chopped meat and reinforced clostridial broth will support growth of this strain. At ATCC, we propagate this item in reinforced clostridial broth in an anaerobe chamber incubator and use 80% nitrogen, 10% hydrogen, 10% carbon dioxide gas mixture at 37 degrees Celsius. Other methods that we will discuss later in the presentation, such as needle and syringe methods and sachets and jars, would be sufficient for this item due to its aerotolerance.
Aerotolerant anaerobes are different from facultative anaerobes in that they cannot use oxygen to generate ATP. Instead, they rely on fermentation. A wide variety of medium, such as PYG, reinforced clostridial broth, TSA with blood agar, and Brucella blood agar may be used. At ATCC, we usually propagate and incubate these organisms in the anaerobe chamber and incubator because they will generally use the 80-10-10 gas mixture and grow at 37 degrees Celsius. However, these organisms can easily be propagated using other methods described later in the presentation. A good example of an aerotolerant anaerobe is Clostridium tertium. It is a gram-positive motile rod with terminal spores. While sporulation occurs readily on most media incubated anaerobically, it does not when incubated aerobically. Because it is aerotolerant, you will probably observe some growth on a blood plate incubated aerobically. It was isolated in 1917 by Captain Henry from war wounds and was the 3rd most common bacteria he found, hence the name tertium. However, it can also be found in soil, feces, infections, and is mainly non-pathogenic. Strict or obligate anaerobes grow only in the absence of oxygen and will be inhibited or killed by oxygen. They generate ATP through anaerobic respiration or fermentation. Cultures can be grown on a wide variety of media provided that it is reduced. Media such as PYG, reinforced clostridial broth, TSA with blood agar, and Brucella blood agar meet a variety of nutritional needs. Using an anaerobe chamber is the ideal method for propagation and incubation. 2 examples of obligate anaerobes are Fusobacterium nucleatum subspecies nucleatum and Clostridium sporogenes. ATCC 25586 is a non-spore-forming, gram-negative, thin, spindle-shaped rod with pointed ends. It was isolated from a cervical facial lesion. Most strains produce hydrogen sulfide and all strains produce butyric acid. It requires a rich media such as Brucella blood agar and chopped meat broth. ATCC 19404 was originally isolated from gas gangrene and is a gram-positive motile rod with subterminal spores. Sporulation occurs readily on most media. It is genetically similar to Clostridium botulinum but does not produce a toxin. For this reason, it is used in research and QC applications. The environmental anaerobe group includes extremophiles, although it should be noted that not all extremophiles are anaerobic. Some examples are thermophiles, which require growth temperatures from 45 degrees Celsius up to 90 degrees Celsius. Halophiles thrive in high salt concentrations, Methanogens produce methane, and alkalophiles require a pH of 8.5 to 11 for optimal growth. Piezophile are microorganisms adapted to the high pressure and cold temperature environment of the deep sea. Many environmental anaerobes have very specific nutritional and environmental needs. They need specific media formulations and will often not grow on agar. Many of these organisms require gas mixtures containing no carbon dioxide. A good example, ATCC 7956, uses mediums such as PYG with tween 80, Lactobacilli MRS agar, and Brucella blood agar. It grows best in a media containing yeast extract and glucose. It has a gram-positive cell wall structure but is known to stain gram-negative. It is a motile rod that grows at 45 degrees Celsius and is known to produce endospores, although they are difficult to observe in culture. It produces ethanol as a byproduct of fermentation. An anaerobe chamber using PRAS media is ideal for inoculation. If the anaerobic chamber is set to a different temperature, the culture must be sealed in an anaerobic jar or bag and transferred to the appropriate incubator.
ATCC acquired the Oregon collection of Methanogens through a donation by Dr. David Boone in 2005. It is comprised of 144 different isolates. Methanogens belong to the domain Archaea and are very strictly anaerobic, with methane always being the product of catabolic metabolism. They are widely distributed in nature, being found in anaerobic habitats such as aquatic sediments and the gastrointestinal tracts of animals. One example of the methanogen group is Methanobrevibacter wolynii, ATCC BAA 1170. Extremophiles such as this have specific growth requirements. This item requires a media redox potential below minus 110 millivolts, which can be acquired by using a reducing agent. Methanobrevibacter also requires a large amount of pressure in the headspace of the test tube. 20 psi of 80% hydrogen and 20% carbon dioxide facilitates growth of this organism when using Belch tubes. Growing cultures may have to be regassed and repressurized during incubation using a needle and syringe method to maintain 20 psi. This item grows in a very intricate medium with numerous components to satisfy complex nutritional requirements. ATCC does not grow this item on agar plates as it is difficult to achieve the pressure and strict anaerobic requirements. The anaerobe chamber is the ideal method for propagation of this organism. If a different incubation temperature or atmosphere is required other than what the anaerobic chamber is set to, the culture must be sealed before being removed from the chamber for those adjustments post-inoculation. Understanding your anaerobe's nutritional requirements is key to selecting the correct media. Berge's manuals are a great resource for this. Overall, commercially available pre-reduced, anaerobically sterilized media is superior because it is autoclaved, dispensed, and packaged anaerobically in light-proof packaging. If the media is made in-house, ensure it is stored in the dark and used within 7 days. If making agar, within a couple hours of pouring the plates, move them into an anaerobic environment, for example, a jar or a chamber. Indicators are highly recommended additives used to monitor the redox potential of the media. Rizazirin is commonly used because it is non-toxic to bacteria and effective at low concentrations. Rizazirin becomes colorless at a redox potential below minus 110 millivolts and remains pink above minus 51 millivolts. Some organisms require redox potentials lower than minus 110 millivolts and may not start to grow even if the medium is colorless. On the other hand, if the organism produces a strong oxidant, such as nitrate, the redox potential may rise above minus 51 millivolts turning the media pink even though it has not been exposed to oxygen. Common bases for blood agar media are Brucella, which is enriched and good for fastidious organisms, Columbia, which is a combination of animal and plant proteins to support fastidious and non-fastidious organisms, tryptic soy, and brain heart infusion 0.5% yeast extract. All of these blood agar bases are non-selective and will support growth for most anaerobes. Some anaerobes may perform better on some bases over others, so again, knowing your anaerobe's nutritional requirements are key to successful growth. If there are problems with achieving growth on agar, choose a medium that has supplements. Selective agars are useful when trying to identify an unknown culture or to separate a mixed culture. One example is BBE agar, which selects for Bacteroides, Bilophila, and Fusobacterium. It is supplemented with gentamicin to inhibit aerobes and bile to inhibit most other anaerobes. Chopped meat broth will support the growth of the vast majority of anaerobes. Reinforced Clostridial broth is non-selective, but is used most commonly for growing the genus Clostridium, along with Bifidobacterium, and the Propionibacterium we discussed earlier. When having growth problems, PYG or supplemented tryptic soy can stimulate growth in anaerobes due to the additions of carbohydrates, yeast extract, hemin, and vitamin K1. At ATCC, we have found that medium 2722 is especially good for growing Porphyromonas. Thioglycolate is common for isolating anaerobes, but it is not sufficient for substantial growth. For good growth in broth media, inoculations should not be too dilute, and subcultures should not be made too late in the growth curve. The two recommended test tube formats for growing anaerobes in broth are Hungate tubes and Balch tubes. Hungate tubes are comprised of a test tube or roll tube with a butyl rubber closure held in place by a screw-on cap with a circular opening for a needle to be injected. Balch tubes are comprised of a test tube, a solid rubber stopper, crimped with an aluminum seal. The latter test tube is ideal for extremophiles as they can sustain pressure and extreme temperatures. Proper media storage is important to prevent inhibitor formation. Inhibitors that affect the quality of the media are oxygen, light, and moisture or dehydration. Oxygen causes the oxidation of organic ingredients, and light creates peroxides. Storing media in the dark and using it within the recommended time frame will prevent inhibitors from damaging the media. Inhibitors that affect cell growth can be due to incorrect media or atmosphere selection, the presence of bile in the medium, using too strong or too weak of a reducing agent, having oxygen present in the medium, using the incorrect anaerobic atmosphere, or failure to add the necessary supplements for an organism, such as hemin, yeast extract, vitamin K1, or tween, can inhibit an organism from growing.
Reducing agents should be used with all anaerobes, but they are particularly important for strict anaerobes. The reducing agent is oxidized by the oxygen in the media, and therefore the media is reduced of the oxygen. This effectively removes the oxygen that can harm the anaerobe. Some reducing agents can perform quickly but generate toxic reaction products like sodium dithionite. Sodium sulfide precipitates essential metal cations and can be fairly toxic to some anaerobes like ruminants. Cysteine has a low toxicity and does not have ancillary chemical reactions, but the rate of reduction is slow, several hours at room temperature. This is the reducing agent that ATCC recommends most often. You should select the reducing agent that is used in the media formulation. It is typically used in a ratio of 2 milliliters of reducing agent to 100 milliliters of media. Allow the reducing agent to react for at least 1 hour, however 12 to 24 hours is preferable. Oxygen is harmful and even toxic to all cells. Cells have several defenses against oxygen toxicity that are listed here. For example, hemoglobin binds molecular oxygen, superoxide dismutase acts against superoxides, catalase acts against hydrogen peroxide, and vitamin C controls free radical chain reactions. The damage to cells caused by oxygen includes oxidation of lipids in membranes, which will affect gram stains, inactivation of enzymes, which will affect characterizations, and direct genetic damage. To answer why is oxygen toxic, we need to look at the reduction of molecular oxygen. Oxygen has two unpaired electrons in the ground state, and they are spinning in the same direction, thus they are not reactive. When energy is added to the ground state, this produces the singlet state, where the electrons spin in opposite directions and are very reactive. The reduction of molecular oxygen produces toxic intermediates. As free molecular oxygen is metabolized, toxic intermediates are produced. Some organisms use superoxide dismutase and catalase to buffer the effects of the superoxide anion and hydrogen peroxide. There is no defense against the hydroperoxy radical. So while all cells suffer some effects of oxygen toxicity, the effects of oxygen on anaerobic organisms can be detrimental. Once the damage is done, it cannot be reversed. The best defense is to prevent exposure to oxygen. This is why it is important to have media completely reduced before inoculating. A number of gas mixtures are available. An anaerobic environment requires removing free molecular oxygen and replacing it with the gas mixture. Most anaerobes can use a range of gas mixes. 80% nitrogen, 10% hydrogen, 10% carbon dioxide, and 80% nitrogen, 20% carbon dioxide are widely used. In our anaerobe chambers, we have replaced 80% nitrogen, 10% hydrogen, 10% carbon dioxide with 90% nitrogen, 5% hydrogen, 5% carbon dioxide to reduce the chance of explosion. For our purposes, these two mixtures are interchangeable. Generally, extremophiles will be the organisms that require a specific mixture, such as 100% nitrogen, 100% methane, or 97% nitrogen, 3% hydrogen. While unlikely, if you are required to use 80% hydrogen, take extra precautions as it is highly flammable.
The roll tube method was developed by Hungate for isolation of oxygen-intolerant anaerobes from ruined fluid and sewage in 1950. It was later modified and improved throughout the 60s and 70s. 4.5 milliliters of pre-reduced agar medium was placed in a Hungate or roll tube and kept melted in a 46-degree water bath. Oxygen-free gas was fed through the sterile leads, at least one which is bent, and attached to a lure lock tip syringe. The ambient air in the syringe was replaced with oxygen-free gas through a lead attached to a modified Pasteur pipette. The syringe was placed into the growing anaerobic culture, the inoculum was removed and placed into the prepared roll tube of melted agar. The tube was then placed on a rolling device that spun the medium until it remained as a solid layer on the wall of the tube. Historically, this method was not widely used as it was hard to perfect the technique. This is an example of a culture growing in a roll tube. The Cannula System is a modification and updated version of the roll tube method. Cannulas are sterilized with a Bunsen burner and attached to an anaerobic gas mixture. The gas mixture is turned and set at the appropriate psi to a gentle stream of gas so that the broth media does not bubble or splatter. The cannulas are inserted into test tubes of media and/or growing culture. The anaerobic gas is supplied in a constant stream to maintain the cultures in an anaerobic environment. Work can be performed in a biological safety cabinet using pipettes for subcultures and inoculations. This method is a good alternative to an anaerobe chamber. Please note all media is pre-reduced with a reducing agent and oxygen-free gas before use in this propagation method. Also, superior aseptic technique is critical to this method. Cannula systems are not readily commercially available and would most likely have to be built in-house. The needle and syringe method is the simplest and cheapest way to culture anaerobes. This method is useful to pre-reduce media and to perform inoculations. To reduce media, first sterilize the rubber stoppers of the test tubes. Then a reducing agent is added and the tubes are outgassed with a line of oxygen-free gas, displacing the air inside the tube through a syringe vent. Removing the vent first ensures no backflow of ambient air into the test tube. Subsequently, to perform inoculations, insert a needle and syringe through the rubber stopper of the test tube and invert the tube to withdraw an inoculum. Insert the needle and syringe into another tube and dispense the inoculum. Plates can be inoculated if the organism is facultative or aerotolerant and placed in jars or bags with a sachet immediately after inoculation. Aseptic technique and the ability to work quickly and safely is critical to this method. Organisms that need to be repressurized or regassed, like ATCC BAA 1170 discussed earlier, would be performed in this manner.
This is an example of a Mcintosh Fields Jar. This method was first published in 1916. Originally, it was a metal jar with an airtight lid and a screw to hold the lid in place. It had a pressure gauge to read internal pressure. After partial evacuation of the jar, an electric current was passed through a catalyst to heat it, which caused the residual oxygen to combine with hydrogen gas to produce water. The manometer measured this as a fall in internal pressure of the jar, thus leaving an anaerobic atmosphere within the jar. Then hydrogen was pumped into the jar so that the pressure of the jar equaled atmospheric pressure. Mcintosh and Field also developed a methylene blue indicator to ensure anaerobiosis. Anaerobe jars evolved over time. Brewers jars were developed in the 1950s and 60s and featured a self-contained, disposable hydrogen carbon dioxide generation system. Hydrogen was produced when water activated a sodium borohydride tablet. The hydrogen then reacted with oxygen and the palladium catalyst present in the lid of the jar. Carbon dioxide was generated and produced a suitable atmosphere for anaerobic growth in about 90 minutes. Newer versions feature a cold catalyst and involve a simple sachet without the need for water and a catalyst. These produce an anaerobic environment in less than 30 minutes. Anaerobic indicators are useful in these systems to ensure the conversion to an anaerobic environment is successful. The Anoxia Mat is an economic alternative to an anaerobe chamber. It is a small self-contained unit that doesn't use a lot of bench space. You can attach more than one gas tank, allowing for the customization of gas mixtures. It automatically evacuates and seals the jar, after which the desired gas mixture is pumped into the jar. The anaerobic atmosphere is immediate as opposed to the jar and sachet method. The Anoxia Mat can also be used for microaerophilic organisms. Anaerobe chambers are the ideal propagation method for anaerobes. Pictured here are the BugBox Plus and the Whitley MG500 anaerobic workstation. Both use foot pedals to gas out the sleeves prior to entering. Items are brought in and out of the chamber through a pass box that is outgassed to prevent oxygen from entering the chamber. Vials can be thawed or rehydrated in the chamber, preventing any oxygen exposure. Both of these models allow the user to set the interior temperature, so they can also be used as incubators. Using an anaerobe chamber allows you to perform all work in an oxygen-free environment. This is the Anaerobe Systems 580 gloveless anaerobe chamber. We ordered ours with the built-in incubator that we keep at 37 degrees Celsius. Having the incubator separate from the workspace is much more comfortable for the user, and the incubator accommodates a large number of plates. Cultures that need another temperature are inoculated in the chamber and then placed in a jar with a gas pack. The jar is taken out through the pass box and placed in the appropriate incubator. Anaerobe Systems recommends using the 90% nitrogen, 5% hydrogen, 5% carbon dioxide gas mixture. When working with an organism that requires a different mixture, we outgas the test tubes using the needle and syringe method. The chamber uses positive pressure to push gas out through the sleeves when you enter and exit, eliminating the need for successively gassing out the sleeves. It also gasses out the pass box faster than other models, thus using less gas. This anaerobe chamber is economically priced and easy to maintain. Most importantly, using an anaerobe chamber keeps your anaerobes in a completely oxygen-free environment, which increases your odds for success.
I would like to thank Ms. Faust and Ms. Kruger for the excellent presentation, and thank you everyone for attending the webinar. Please join us for more webinars in the ATCC Excellence in Research webinar series. Thank you again and have a great day.